Hi, welcome back to the Sound of Science. Today we are going to be talking about the cell cycle and how it's regulated. This is AP Biotopics 4.6 through 4.7. You absolutely need to know the cell cycle. And so, in fact, I encourage you to copy down this diagram that I have sitting to the right. The cell cycle itself is highly regulated. It's made to allow organisms to grow, and it allows, oh, there goes the cat. That's Garfield, in case anybody is curious. The cell cycle itself is designed to allow organisms to grow, for tissue to be repaired, and for some unicellular organisms actually to reproduce. There are two processes that we talk about, interphase and M phase. Interphase is all about growing and preparing and just doing normal cell things. M phase, mitosis, is all about the division of the nucleus. Interphase is split also into three phases. Gap phase 1, which is cell growth. S phase, where the DNA is copied, that's the synthesis phase. And G2 or gap phase 2 is cell growth, where the cytoplasm is doubled and the cell itself prepares to divide just prior to mitosis. In the cell cycle, part of that regulation is checkpoints. There's actually three checkpoints within the cell cycle, and you'll see them denoted with little stop signs. At the end of G1, there's a check that determines if the cell has received the correct nutrients, if it has grown enough, and if there's any damage to the DNA so far. If all is well, it will proceed forward through S phase and ultimately into G2. Towards the end of the G2 phase, we're going to have another checkpoint where we're going to check again to make sure the DNA was copied in the S phase and that there's no damage that we can find. And then finally, about halfway through mitosis, there's going to be an M spindle checkpoint that's checking the spindle fibers attachments to the chromosomes. If these checkpoints aren't found to be the way they need to be, if the cell is not the way it needs to be at this point, then other things can happen to the cell. So, how do these checkpoints work? Well, essentially, the checkpoints are going to work by something called cyclins and cyclin dependent kinases. So, cyclins are just a group of related proteins. They're associated with specific phases of the cell cycle. The concentration of various cyclins will fluctuate during the cell cycle and promote cell progression or the cell cycle progression. If the cyclids are degraded, it inhibits cell cycle progression. The cyclins themselves are going to be specific to their CDKs. Cyclin-dependent kinases. These are a group of enzymes involved in cell cycle regulation. They require a cyclin binding to be activated, and they also are going to phosphorylate the substrates, which will promote certain cell cycle activities. I know this is a lot, but think back in your mind to enzymes and how enzymes work, right? Enzymes bind to specific items, sometimes proteins, right? And when we talk about cell signals, it's the same idea. It's very, very specific. It'll bind specifically because not only does it fit, but it also can be associated with that due to um, whatever charge is there, whether it's polar or nonpolar. So once they bind together, it actually can activate it to move on through the cell cycle. Its presence is a signal to the cell to continue. If it's not there, if it's absent, then that prevents the cell from moving forward in the cell cycle, and ultimately the cell will die. So here on this page, we're going to continue with the how cyclins work topic. So here is an example of what might be happening with these cyclins. So these cyclins have been sort of split into the phases that they're associated with. So there's several different types of cyclin. So you have the cyclin that's found in G1, and it really ramps up and then eventually is going to degrade. G1 tends to last the entire time, the entire cell cycle. The G1 slash S cyclin, however, is going to ramp up right at the end of the G1 phase, and then it's going to fall off sharply about halfway through the S phase. The S cyclin ramps up starting in the S phase, and it's going to fall off before the M phase. And the same thing with the M cyclin. It's going to do the same idea. If the protein that regulates the cycles are damaged, then the cell cycle is not regulated. This is just like any other mutation, except in this case, if one of these proteins is not regulated appropriately, you're going to end up with a cell that literally doesn't know when to stop. And it will just keep dividing through uncontrolled cell division. Let's say that the cell doesn't pass the checkpoint, that there's a problem. And if the cell recognizes that there's a problem, then what might end up happening is something called apoptosis, which is programmed cell death. If the cell doesn't recognize the problem, primarily because the problem may be in that protein, one of those cyclin proteins, then ultimately it's going to continue to go, even though there's mistakes. And instead of becoming a normal cell, then in that case, that cell is going to become a cancerous cell. And that cancerous cell is going to continue to divide because the cyclins are not being produced to stop it. So it will divide fairly constantly. And that is how we get tumors, and that's how we get cancerous tumors. So we're going to continue now through M phase, mitosis. M phase just ensures a complete transfer of the genome from a parent cell to two identical daughter cells. Mitosis has 4 phases: prophase, metaphase, anaphase, and telophase, which are listed here. If you don't recognize these, you need to make sure that you do. Draw these out, label them. In the next couple of slides, we're going to talk about what happens in each of these phases. So make sure that you are leaving some space in your notes to describe what's happening as well as to draw it. Prophase is the first phase of mitosis. The nuclear envelope begins to disappear, and the DNA coils into chromosomes, as well as spindle fibers begin to show up and move towards the center of the cell. Prophase is really the phase of first, which I always think, pros are always number one, right? So this is the first time the nuclear envelope is going to disappear. This is the first time the DNA is going to coil up into these chromosomes. And this is the first time we're going to see these spindle fibers. So it's a lot of first happening. Sometimes the start of prophase, you're catching that nuclear membrane starting to fall apart. It's really easy to identify. In the metaphase, fibers, the spindle fibers, are actually going to align the double chromosomes across the center of the cell. They line up in the middle. Metaphase has always stood for middle for me. So metaphase, they line up in the middle. Prophase, lots of first, the first time we see chromosomes. Metaphase, middle. That's where all your chromosomes are. And I want you to notice when you look at metaphase that the part where the spindle fibers attach is actually called the centromere. That's the center where the two sides of the chromosomes, the two chromatids, actually attach to each other in the center. That's called centromere. The centriole, which is part of animal cells, is actually controlling those spindle fibers and moving them throughout the cells to make sure that they're where they're supposed to be. During anaphase, these spindle fibers are actually going to start pulling the chromosomes away from each other. For me, anaphase, I always think of away. Anaphase means away. And so what you'll start to see is that your cell is going to start to elongate, start to become more oval, less round, and you're going to start to see your chromatids separate from one another and move to opposite sides of the cell. Anaphase, away. Finally, we have telophase. Telophase comes at the end. And when you look at telophase, you are starting to see the development of 2 nuclei. The nuclear membrane is about to reform. Each nucleus will contain a complete genome, which means a complete set of chromosomes. And those chromosomes are going to start to uncoil. They're going to start to look more like chromatin again, which is where DNA kind of looks like a hairball. So telophase to me, I always think telo means the end. And if you look at telophase and you look at the cells, they're sort of like this and kind of look like a rear end, which is how I remember telophase. Or you might remember that telophase has a T in it, just like the word but. Finally, we have come to the last part of the cell cycle before we start over again, and that's cytokinesis. Cytokinesis means division of the cytoplasm. This is where the cell divides completely into two identical cells. So this is where we have an equal distribution of organelles in cytoplasm. The nucleus has already divided. It finished doing that in telophase, which would signal the end of the M phase. Cytokinesis is sort of part of mitosis, but not really part of mitosis. But when the cell completely separates and moves away from the other identical cell, now each of these is free to start the process over again, going back through G1, unless the cell pops into G0, which is sort of a holding phase. So G0 is sort of outside of the cell cycle. This is where cells go just to honestly live their best cell life. They're not really doing a whole lot. They're just being cells, doing cell things. If the signal is received that it's time to divide, like let's say that there's some type of injury or growth hormone is being produced at a rapid rate, then that can actually indicate to the cell that it's time to go back through the cell cycle. And so at that point, it'll actually re enter G1 and continue where it needs to go. This is the end of our video for tonight. Hopefully that was quick enough for you. As always, thanks for watching, and I'll catch you next time on The Sound of Science.